Good afternoon and welcome to At Yale Live. I'm Eric Gershon. Today we talk about a life in science with a man who's lived it to the fullest. James E. Rothman, professor and chair of the Yale School of Medicine's Department of Cell Biology and a winner of the 2013 Nobel Prize in Physiology or Medicine. As always, we'll take some of your questions. Please submit them via Twitter to @Yale or by email to socialmedia@yale.edu. Jim Rothman, thanks for being with us.
Pleasure, Eric.
So on the day that you won the Nobel Prize, you taught all your scheduled classes. Tell us a little bit about why that was important.
Well, teaching is an important obligation. And I think I was trying to send a message to the students and also my colleagues that, we should keep our focus on the most substantial things. Teaching is a very substantial thing. Research is a substantial thing. And, it was wonderful to have received an award like this, but it's in recognition, we hope, of substantial things, so let's remember the substantial things.
Tell us a little bit about the Nobel ceremony and the experience and all the hoopla surrounding that.
Oh, no, that's a really big question because it's an experience I would recommend to anybody. But to be serious, it's like stepping into another world, and it's quite an unreal experience. The Swedish, one of the things you learn is that the Nobel Prize for the Swedish people has a significance above and beyond, by far, what it does internationally. For them, it's almost like a national celebration. It comes at the time of the year when there is the least light in a country that is in Scandinavia and is dark in the winter, and so it's made into a festival of light, and it's become a very important part of the Swedish brand and their brand equity, if you want to think about it in economic terms and political terms. So there's the level of celebration is really quite extraordinary.
Was there an opportunity to mingle with laureates in other disciplines, or were you mainly with your fellow scientists?
Well, both, actually.
Yeah.
And so there are quite a number of, all the official events involved all of the Nobel laureates, except for the Nobel Peace Prize, which is awarded in Oslo. Mm-hmm. All of the other ones are awarded in Stockholm. And so you spend actually quite a bit of time getting to know some interesting new folks and their families, and and actually some of the children became friendly, and I'm told that my 3-year-old granddaughter was quite friendly with Eugene Fama's 3-year-old granddaughter, and so, there are relationships yet to come. Well,
that's an amusing anecdote that I think we both understand, but Eugene Fama shared the Nobel Prize in the economic sciences with Bob Schiller.
That's right.
Who was on the faculty here at Yale.
That's
also.
Right.
Had you met Bob before? Or?
Well, actually, the first time I met Bob was right in the studio. I mean, Yale is a
here.
Yeah, I mean, Yale is a big place,
yeah.
And we're in different faculties, and I've only been here for 5 years.
Yeah.
But I have to say that Bob and I became quite friendly, and continue to be friendly, mm-hmm. and as do our families. Yep. And so out of this will come a very good, and fun relationship. We actually continue to send each other little notes about how's it going. And sharing a few hints about how to survive as a Nobel laureate.
Bob was telling us that he had to, and he was joking, but he declared email bankruptcy after he won the Nobel Prize because he was so inundated with email, congratulatory, people asking him to do things, so on and so forth.
Yeah.
And he felt that he should respond in some way to each of them, but he couldn't possibly. I don't know if you had a similar sort of experience.
Well, I would never trust an economist who claims he's bankrupt. Of course, you get a lot of email.
Yeah.
I tried my very best to answer all of the email that came in the early days. But there are a lot of requests. Some of them are kind of amusing, but and I won't reiterate them here. But for the most part, what happens is that life gets back more towards normal, which is a good thing.
Let's talk about science a little bit.
Yeah.
On the day that you won the prize, you gave a very eloquent layman's description of the body of work that led to the prize. Could you reprise that again for our audience?
I can try.
Yeah.
I can try. So the The particular piece of work, I mean, all scientists have a broad range of things that they've done, but I think the particular aspect of our work that was that the Nobel Committee focused on has to do with cell-to-cell communication in the body, and it really relates to how cells organize themselves within each cell, and how the cells are organized and maintain a sort of synchrony of behavior and in life. Okay, so that sounds very general, and in a way it is, but to try to make it a little bit more particular, for example, all the cells in our liver, just to pick a part of the body, each one has to, metabolize glucose properly, otherwise you have diabetes. Mm-hmm. But they also have to communicate with each other so that they all behave in a similar way. Now, those cells behave differently than the cells in your skin or in the many types of cells in your brain. And so in order to maintain the communication between the cells in an organ or between the organs in a body, so that the entire body functions as we know ourselves to function, as an intact entity, there are molecules that are released by cells that actually provide for the language. Mm-hmm. And a liver cell will secrete molecules that go into the blood and affect the behavior of other cells in the body, and even within the cell. There is a high degree of organization of the processes in the cell. We call it compartmental organization in the field of cell biology. Mm-hmm. And these various compartments also need to communicate with each other. And this is all done by a process of, underlying all of this is a process called vesicle trafficking, which is what the Nobel Prize mm-hmm. recognized. And really what it is that inside each of our cells, there are many tens of thousands of little vesicles. They're like cells within the cell. They're very, very tiny. And they have the shape of bubbles. They're little spherical, sort of bubble-like particles. And each bubble sequesters within it a packet of proteins and other molecules. Mm-hmm. And depending on the nature of the bubble can release its content to the outside of the cell so that content gets released and then signals cells that are nearby, or maybe the content in other cases gets into the blood and signals to an organ at a great distance. Or maybe sometimes the bubble or the vesicle rather than targeting to be released from the cell, targets some part within the cell and therefore delivers its cargo there and passes a signal in that respect. So in that, there's an underlying framework of what's called membrane fusion. That's the technical term for how a vesicle or a bubble merges with its target to deliver the cargo, and it gets very technical mm-hmm. and arcane. But the beauty of it is that there's an underlying, very sort of simple biophysical principle that accounts for this enormous diversity of physiology that allows us to function as an integrated whole. And when you have imbalances in these various processes, you get diseases, like type 2 diabetes. Mm-hmm. There are imbalances in neurotransmitters in most common psychiatric disorders, probably in neurodegenerative disorders. And there's a long list of unfortunately, very common diseases in which these pathways are altered. So I think the combination of the sort of the very basic principle that's involved and the kind of generality is what, and the impact on understanding human disease is probably what the Nobel Committee was trying to recognize in our work and that of my colleagues Randy Sheckman and Thomas Suthoff.
I recall from October that you were asked, was there a eureka moment for you, a single one? I imagine perhaps there were many, but if I remember correctly, you said, well, that's not really how science works. Talk to us a little bit about the nature of scientific discovery.
I'd say not a bad analogy would be playing baseball. Okay, so even if you're an above-average hitter, you got to go to bat a lot. Okay, and so you have to be swinging, and so you have to pay attention, and so you have to do that. But there's another thing that you don't normally think about with baseball, which is that imagine the batter isn't inside the stadium. He can swing all day, and nothing will happen, right? So you got to be in the right place, meaning in the stadium. You got to be trying pretty hard. Okay. Now, Thomas Edison used to say that inventorship is 99% perspiration, 1% inspiration. It's a very famous kind of one of his more famous quotes. And that's probably true to some extent for scientific discovery. So the perspiration is swinging away every day in the lab, in the baseball field, in the stadium. But then how do you know that you're in the stadium, okay? How do you know that you're in that place, okay? That actually takes a lot of judgment and skill, intuition, and it's not 100% clear where that comes from. But that's the piece that can come in a eureka moment. Oh, you know, I really should be doing more of this than that. Maybe I should be looking at this rather than that. And it may take a year, it may take 10 or 20 years to exercise that thought. And you probably have to go to bat a lot of times. You'll make a few mistakes. You're certainly going to be thrown out quite a bit. Okay, usually you get to walk instead of getting a home run. But if you keep at it long enough, and what probably distinguishes the best scientists from those who are not as fortunate or perhaps don't have as much skill, is the ability to be more often in the baseball parks when they're swinging, rather than swinging it, into the thin air.
So I was going to ask this later in the conversation, but I think it's appropriate now. What has been your strategy for picking risks? Because a decision, right, to focus on an entire lab and the resources of that lab for a year, perhaps longer, is necessary, but it's a risk. So how have you approached that historically?
Yeah, well, Eric, that's really, these are really good questions. I think one of the most fundamental things is being willing to take a risk. Okay, now most people actually, even scientists, are not ultimately prepared to take a risk. Okay, and these days in science, maybe a subject we can touch on again later, it's actually massively more difficult to take an appropriate risk. There's a kind of judgment that comes into it, right? So you have to feel, well, I'll tell you when I was starting out, okay?
Sure.
I was really fortunate. My first job as a young professor was at Stanford University in the Department of Biochemistry. And the great figure at the time at Stanford, and indeed one of the greatest biochemists of the last century, was a man named Arthur Kornberg.
Okay.
So, in fact, I went to Stanford because I was going to be able to have my lab
to teach,
there.
Right?
Or to do research, and teaching is a part of it. Okay. So, and Arthur had received the Nobel Prize in the late 1950s for discovering how chromosomes are copied in cell division, essentially, an early version of that. And at the time, what he did was just extremely remarkable. The biochemistry he did, no one else could have imagined that it would have worked at the time. So I said, Arthur, how did you decide? Just the question that you asked me. And he said, well, Jim, something like this. You know, I thought I'd try to do something that nobody else thought could be done, but which I thought could be done, and that was really worth the trouble. Now, there's a lot of, that's an easy formula to lay out. It's not actually an easy formula to
carry out.
Okay? And there's probably some luck in it, but, if you're self-critical and you wake up every day and you ask yourself, am I doing the most important thing that I could do? And is it really unique enough? Then you will make discoveries, and that's the power of the scientific method.
I'm going to take a question from a viewer. This one comes in by email from Karen Rasico, who asks, what is the most critical advice you give to your students / postdocs as they move into their first independent positions?
Okay. Well, Karen, that's...
Please go ahead.
Oh, I'm sorry. Karen, that's a really interesting question. You'll have to ask my students and postdocs to find out if I'm telling you the truth, because sometimes I don't always remember it right. But my feeling is that that you need to keep working in the lab yourself. And I tell this all the time to maybe more importantly when we have a new assistant professor at Yale and they come and ask for my advice the way I did Arthur's.
Yeah.
I went to Arthur Kornberg. What I say is, look, the best person in your lab is you. And as a matter of fact, you're the only person in your lab. So your best asset is you. Work in the lab. Keep your focus on working in the lab. And even as your laboratory grows, make sure that you yourself work with your own hands. And I followed that recipe for the first 10 years of my career, and a great many of the experiments that turned out to be important, I didn't do them all myself, but some of them I did myself. And the ones that I didn't do myself entirely, I did some piece of. So, you know, there's a kind of intellectual investment that you have through your own activity. And so that's actually my single most important piece of advice. And the second one is what you, Eric, touched on, which is take a risk. Yeah. And what I tell the assistant professors are, look, you know, you're coming here and to a great place, Yale University. There are other great places. But you have the resources now for a couple of years to get started and to do something original. And, you know, you should do it. And if you take too conservative approach, you may succeed in the sense that you survive, in the sense of Darwin's finch. But, you know, did you go into science to become Darwin's finch and to find a niche? I certainly hope not, because if you did, you know, please find another job. Okay?
Let me take another question. This one by email from Janet Lyons. And this is also a broad question. She asks, what areas offer the greatest opportunity for basic research today? And what areas offer the greatest opportunity for tech transfer? I imagine different scientists would have different responses, but I'm curious to hear yours.
Boy, that's really tough. If I, you know, this is, I think that this might be another version of the question, what would you do if you were starting out again? Okay, and so it's one that I'd like to address. The first thing I would say, is it Lyon?
Janet, yeah.
Janet, sorry,
Lyons, yeah.
Janet. The first thing I would say, Janet, is don't ever listen to somebody like me when you ask a question like that, okay? And I mean that in absolute seriousness, because as an older scientist who is matured and been well recognized and so on, I'm the least likely to succeed if I were to start all over again. So my judgment is to be immediately suspect. Nevertheless, you asked the question. So in basic science, I think that absolutely the most open area is the brain. But not necessarily in the way the brain is commonly approached. The common approaches to the brain, more and more anatomy-driven, as they always have been. You know, there's President Obama's project to, you know, measure all of the connections in the brain and work out all the anatomy and so on. There is sort of artificial intelligence approaches to the brain. There are very cellular and molecular approaches to the brain. They're all very good. But I think they kind of miss the point at some level of asking some simple questions about the brain. What I find amazing about the brain is that all brains have fundamentally the same anatomy of the cortex. They have 6 layers. I tend to think of the brain as a repeat structure of with a common cellular wiring, and every portion of the cortex can be used, it appears, to do any job. It's just a matter of what its input and output are. Well, that's easy to say. What is the underlying principle that explains that circuit? Now, there are people who are trying to do that, but there isn't, in my opinion, anywhere near enough focus on that question. So whatever it is, I think the understanding of what allows mind to emerge from matter is the single biggest question of our time. I'm a little skeptical as to whether in our lifetimes we'll understand it, but I'm not skeptical that in the long run we will. Now, technology transfer.
Which maybe we should define for viewers, is essentially the creation of technologies and companies that make or those technologies that comes out of university research. Is that a fair description?
Yeah, that's a fair description. I'd like to redefine the question, though, slightly. The first question clearly revolved around basic science, but let me just say, let's say, what could emerge from engineering? Because engineering is typically engineering is the use of scientific principles for new purposes as distinct from the discovery of new scientific principles. And engineering is an extraordinarily vital area. And the area that I know much more about is biomedically related engineering. And I am impressed by the potential for the interface of machine and body. You know, I think that we're learning as we learn more about the brain, for example, and other parts of the body, as we develop more materials on the nanoscale, and we develop the ability to manufacture on the nanoscale interfaces between body and inorganic matter, we will begin to see a merger of, I don't mean to sound like a science fiction guy because I don't regard this as science fiction, we'll begin to see more of a kind of gradual coming together of machine and man, first in limited ways. It's going to take off first, I believe, in the commercial market with on-body sensors, which will come out of health enthusiasts and the need for home health and healthcare reform, which is going to drive stuff out of clinics and more towards the home and less sophisticated interrogation by, let's say, experts rather than doctors. Mm-hmm. There's going to be, I think, a whole series of events that are going to provide the commercial driver that essentially focuses invention where there's a need. Mm. And so I think that's, if you want to call it tech transfer, you could call it that. I don't know if it's going to come out of the universities or whether it'll come out of the startup companies or some combination of the two, or maybe it won't even come from the U.S. I don't know where it's going to come from, but the world 50 years from now, I think, is going to be really different in that regard.
Fascinating. Another question, this one via Facebook from Peter Cohen, and this is a slightly more technical question, but I'm sure you'll be able to handle it. Do you think that a greater understanding of path mapping in 4D could be useful in the sciences?
I'm not sure what we mean by, I don't know if we have a chance to feed back to the questioners, but if path mapping in 4D, okay, no, I'm going to interpret that. I'm hoping I get this right.
Yeah, sure.
I'm going to interpret that to mean the fourth dimension as being time. So ordinarily when we talk about 4D, in most situations, we mean 3 dimensions of space and 1 of time. So if that's the case, I mean, I think we need to understand certainly physiology, in the time domain. Mm-hmm. And another, in my field, I've been speaking up until now about most of the things I don't know about, which is something I'm asked to do more and more these days, and that's another health warning. But in the field of cell biology and cell physiology, we have new imaging tools that are really astonishing now. And the ability to observe the biochemical and physiological pathways even within the cell at unprecedented resolution and simultaneously in real time is beginning to give us just the beginnings of an understanding of how a cell functions as a computer. Wow. Okay? Because I know that people don't normally think about it this way, but something that you might think about is that, in electronics, there's something called an analog to digital converter, okay? And it takes an analog signal and it converts it into a digital all or none outcome. Now, I think we understand that computers can do that, but most people don't appreciate that cells also do that, as do organisms, right? So, for example, a cell, every moment of its life is making a decision. Do I continue as I am, or do I decide to divide and become 2? That's an all or none decision. You're absolutely committed to divide when you divide. Or perhaps you decide to move around rather than stay where you are, okay? And when that goes wrong, you have a metastasis and a cancer, okay? Physiologically, though, you need it for healing a wound, to bring in new cells into a damaged area. Maybe the cell is damaged because it's for one or another reason, and it decides to commit suicide as a kind of altruistic gesture for the cells in the community around it, okay? Now, all of these are binary decisions, but they're not made based on somebody throwing a binary switch. The cell makes up its own mind autonomously, and the environment contains many signals. Now, you remember I talked to you about signals that are released from the cell for communication. These are the very signals that I'm talking about. They have to also be perceived by a cell. And when you're surrounded by an analog universe of chemicals, you can have a higher or lower concentration of a signaling molecule or another one. That's an analog signal. And yet, that kind of collage of signals needs to be interpreted to come up with an all or none outcome. That's analog to digital conversion. That's a computation that the cells are doing. Each cell does it all the time. We actually don't have the foggiest idea how that works. And understanding what I believe the questioner meant by 4D is probably very important in doing that. And by the way, I'm guessing that type of computation is not so totally different than what the brain does at a multicellular level. It's interesting to think of a single cell as a computer.
A couple questions back, I think we were discussing the question, and you said that another way to think about it was what might you have done in science instead of what you pursued. If I'm not mistaken, you, as an undergraduate, started with math and physics.
That's right.
And then found your way to biology.
Mm-hmm.
Tell me about that a little bit.
Oh, yeah. So I made the mistake when I was at Yale
As an undergraduate.
As an undergraduate, to take a biology course, and it just ruined my whole life, because I became a biologist. I'm kidding, of course. I had intended to be a theoretical physicist when I came to Yale. I can't even remember not wanting to do that, which is kind of a strange thing when you think about it for a kid. And as a result of hearing just an amazing lecture, and my dad was a physician, and he insisted that, in a general way, that I should really at least have a look at biology. At the time, physicists weren't well employed, and I don't think he ever thought that I would really cut it as a physicist. He may well have been right, by the way. And so he suggested I take a course in biology, and I have to say I was completely resistant to the idea, because the first thing you're taught as a physicist is that physicists are really smart. Second thing you're taught is that mathematicians are even smarter. Okay. Third thing you're taught actually is that chemists, well, they're not really very smart, but at least they do worthwhile things and they're respectable. They may be dull. Okay. But biologists, they are really stupid. So, you are primed as a physics student not to ever expose yourself to biologists on the fear of being contaminated. But anyway, I somehow did, and that was just a turnaround. And the reason was very simple, which is that that in physics, it's a very well-developed subject, and even if you're an advanced student, you're so far from the research frontier. It's such a multi-layered subject. Whereas in biology, there's so many questions. And as much as we learn, it seems like there are more questions. And it's not like in physics, with the string this and the string that and the subatomic this and that. And, I had a chance, speaking of meeting the other laureates, had a chance to talk to Mr. Higgs of the Higgs boson.
Wow.
It's fantastic. And I still don't know what a Higgs boson is, except it's obviously important to somebody. Okay, and I don't mean to make fun of it, but, it's just even, intelligent people who really, it's just, you can't get away from the math. And so, and it is very abstract. And so, but biology is, in molecular biology, it's very concrete.
You touched a little bit earlier in the conversation on the question of how it was to be a young scientist when you were starting out.
Yeah.
I'd like to ask you, now you're teaching the future scientists and working alongside them. How is it different being a young professional academic scientist today than it was earlier in your career?
It's very sad, actually. It's massively different. Massively different. When I was, it's different in a lot of different ways. Okay, when I started my lab in 1978. Okay, in that era, until maybe 10 years ago, I would say things started changing about then in a meaningful way. I felt free. Okay, I felt that I felt free to study something of fundamental significance because it was fundamentally important, and ultimately it was recognized to be so. I've been working on that project from the very beginning, the project that led to the Nobel Prize. But I couldn't have justified it other than the fact that it was just something that was fundamentally important. It was obviously important because it was true for all of life, but we didn't understand it. I felt free. I didn't feel under a lot of pressure. I felt pressured, self-generated pressure to learn more quickly, to discover as much as I could. But I felt I had time to fully develop a relatively profound understanding of a subject. Free is the main word. Today, I think the young scientists that I know, they feel confined, okay? And I'm putting it into a black or white box. It's not that simple. Yeah. But even at the best universities, the young scientists today are confined. First of all, they're confined because we give them enough money, we afford them enough resources, private philanthropy typically, to have 2 or maybe even 3 years to get a good start. I had 5 years, so I felt free, they feel confined. I started out with my first research grant at about the time that I started out with a grant when I started my lab. Probability of getting a grant if I wrote out an application for anyone, not just me, was about 40 or 50% success rate.
Oh, we're talking about federal grants, for example,
federal grants
NIH,
from the
NSF.
NIH, National Institute of Health. Chances of getting a grant if you wrote it out was about 40, 50%, and that was on the first attempt, okay? These days, the chances are about 10%, okay, sometimes less, okay, and you get 2 chances at it. So the new assistant professors come without a grant. It's gotten so difficult that they really are not even in a position to obtain a grant for 2 years, and by then they're on the edge of running out of money. They're feeling confined. They're feeling pressured. And the result of that is it forces them to do work that's much more incremental and and involves less risk, much less risk, okay? The result of that societally is that we are already much less competitive globally than we were. And this will continue unless the situation changes. And how can it change? I sensed that question coming. You know, I don't know that I have the only recipe, but the basic issue, of course, the best thing would be to have, as we've always had in medical research, more. But there's a limit to asking for more. And, you know, I don't think it's about asking for more. I don't think we should be going to Congress saying we need to double the NIH budget. It would be nice, by the way, to have an increase rather than a decrease. That is true. But a modest single-digit, sustainable, continuous level of support would be very helpful. But I think that much of the problem that we're experiencing today is of the scientific community's own making. It's not that I personally feel the government, on the whole, over the last many decades, has been there for science in a strong and a sustained way. Yes, over the last several years, we've had issues that have been disruptive, like the sequester, and yes, the NIH budget has been challenged and so on. But I still feel that that broadly speaking, both of our political parties are deeply supportive of sustained biomedical research. I feel that the larger issue is how we allocate the funds within our own family budget. And that hasn't really been addressed in a serious way.
Now, when you say the family budget, are you referring to within
the
universities
NIH
themselves? Within the NIH?
The
okay.
NIH budget has progressively, over the last decade, gone more and more of it is being spent on top-down science projects, research projects. More and more of it is being spent on applied science, so-called translational research, and therefore, since it's a fixed sum, less and less on fundamental research. And it is probably true, in fact, it's undoubtedly true, that 10 years ago, we were not spending enough on translational research so that the public, and all of us being the public, get ultimately what we should be getting from this basic research, okay? But the pendulum may have swung a little far to the point where the, you know, the goose that lays the golden egg isn't laying anymore, which is what I've been describing. And we require fundamental research. And the other is that much of the research is programmatic. So in other words, in the past, it was so-called investigator research that was primarily what was funded. In other words, a young Jim Rothman or young Randy Sheckman starting their lab has an idea, and it's reviewed on the merits of the scientific idea, and it's not put through a filter of what some big guy at the NIH felt was an important program, okay? Remember my health warnings about you shouldn't listen to older people when it comes to, that applies to the people running the NIH too. And you know, it depends on how you tally it. No one knows the exact numbers because the NIH, frankly, isn't that transparent on the numbers. But I've seen numbers to suggest that as much as half of the research is essentially top-down, if you include so-called investigator-initiated applications that are within the rubric of special topics that the NIH has requested. That's a sea change.
Now, as you mentioned, the major problem in your view isn't that there's simply too little money and that the government, for example, hasn't been supportive, but that they're allocating it in ways that you might question.
There is too little money for the end user, so to speak, the person for the researcher.
Okay.
There is too little money for a research community of the size that we have. That is true.
Got it. Okay. So,
okay. And part of the and we need to restructure in the broadest possible way for having. We've been through an exponential phase, broadly speaking, and we're now moving into a steady state. What the government's responsibility is, in my opinion, is to allow steady state growth in a responsible way so that we don't have yo-yos in budgets. You can't have projects that go, you can't have government funding that yo-yos up and down every 3 to 5 years and have 10 or 20 year projects. which you need in any of these areas of research, including translational research. That's the one. But the other responsibilities fall on the NIH itself to closely scrutinize, preferably with outside analysts
Reviewers, yeah.
and outside views, how they allocate their money among the different bins to which it could be allocated. Fundamental research, applied research, top-down, for which there's some value, and bottom-up, just good ideas springing up from young and brilliant scientists. And the third party are universities like Yale. Because as you get to a steady state, we have to change the funding formula to recognize that. We have to keep steady at the wheel, that's the government, okay? And then we have to look at how we structure ourselves as universities. Because we actually have, broadly speaking, I'm not referring to Yale, but broadly speaking, probably too many participants in the research process to be sustained by any model of funding within the constraints of a flat, of a growing, progressively growing, but responsibly growing NIH budget. And the universities as a whole have not responsibly addressed this. So, you know, we really are challenged. And it's not just fair to point the finger at the government. The government hasn't done its bit for the last couple of years, that's true. And the NIH, I don't believe, has been truly responsible in the way that it's dealt the hand of going from exponential growth to flat or steady, has not dealt with that as responsibly as it should. But we as the universities also have not been proactive or sufficiently forthright, in my opinion.
Is there a role that philanthropists can play, and perhaps the private sector, the for-profit corporations in funding science that maybe they're doing somewhat now but could be doing more of? And is that a good idea?
Well, I mean, of course, there is a major role, Eric, even now for philanthropy. In fact, the major philanthropic organization in the United States is the Howard Hughes Medical Institution.
Right,
that is the major one focused on biomedical research. And I shudder to think what America's competitive position in the world would be today had there not been the Howard Hughes Medical Institute over the last 10 years. Now, it's true that only a minority of biomedical research in the U.S. is paid for by HHMI, as it's abbreviated. But actually, if not the majority, certainly a very substantial portion of the truly impactful research is paid for by HHMI, an outsized proportion by probably an order of magnitude. And you have to ask yourself, why is that? Okay? They have a funding formula which is radically different from the NIH. What the NIH funds are individual projects that are reviewed on a forward-looking basis. In other words, I say I have an idea, and here's how I'm going to go about doing it, and some folks on a committee who never meet me personally, or somebody else, I don't mean to personalize it.
Yeah, sure.
Will say, oh, I think that will work, or I don't think that will work, or it's not worth doing, whatever. Now, the Howard Hughes takes a different view. They look at what you've done for the last 5 or 10 years, and they say, did I like that? Was that impactful? And they don't look too carefully at what you're going to do for the next 5 or 10 years. They look at what you've actually done. Now, people criticize Howard Hughes. They say, yeah, but you're only investing in people who have already succeeded. Okay, that's fine. So maybe the NIH and the Howard Hughes need to kind of get their act together a little bit, and the NIH needs to find a way to invest in people who have succeeded, but invest in a different way, as if they were not just out of the gate, and then marshal their resources in a particular way, and they are trying to do that for the younger investigators, but maybe more of that. So there is a huge role for philanthropy, okay, and it is there today. Now, having said that, I think it's great to have philanthropy to help, but even philanthropy tends to be focused on curing specific diseases. And I think that's very important, and it will continue to be very important. But in the main, I don't see that as the solution to maintaining our competitive edge in technology in the broadest sense, which has always come and will always come from basic science and basic engineering.
On the day that you won the Nobel, you were speaking to an audience here at Yale, and you related a little anecdote. I guess you had met with your research group earlier that morning, and you said, guys, it's not over yet. meaning don't slack off. There's more to do.
Yeah.
What, having done so much already, what do you still want to achieve in science, you personally?
Well, there are questions that, a scientist in my position actually, put this Nobel Prize aside, because to me it's sort of an epiphenomenon in your question. It's hard to develop it's almost inconceivable to imagine having an impact in a major way in developing a scientific discovery in a short period of time. So I'm 63 years old. I don't know if I can think of very few impactful pieces of work that take less than 10 to 20 years. So I don't think I have the runway to do that all over again as I did. I'd like to think I have the intellect, but I don't think I have the runway. Okay? So what I'm focusing on is try to go deeper in the questions that I've unearthed. And so, what we're trying to understand, we've taken the process of, let's say, vesicle fusion, how the bubbles target and release their cargo for signaling and other other biological events. And we've now learned how to we haven't just in the past we discovered how it works at a certain level. Now we want to understand it at the level of atomic detail. So we're and so that's a process that we're very much engaged in. There is in the connection of really finishing off a piece of work, there's one very major question that is not yet understood appropriately that relates to communication in the brain. So when in the brain, we have these neural networks of neurons that have to talk to each other, and one guy fires an electrical signal and it needs to transmit the electrical signal to the next neuron in order to enable a neural computation to take place. That's ultimately what cognition is about, played out on a large scale. That's called synaptic transmission. And at the end of every nerve, there are many little bubbles called synaptic vesicles that store the neurotransmitters. Same idea as what I said before, but a very particular version of it. And when the electrical impulse comes to the end of the wire, the nerve axon, as it's called, the neurotransmitter is released, and it passes the signal to the next nerve to which that nerve is wired. Now the neurotransmitter has to sit in these little vesicles waiting for the electrical signal. How does it know exactly when to release? It's not how it gets released, that's the general problem. How does it know when to release? If we didn't release our neurotransmitters at the right time and at the right place, our brain would be nothing but a confused fog, okay? So timing is everything in the brain, absolutely everything. And my colleague, Tom Sutoff, with whom this award was shared, well, Tom discovered the protein machinery that controls the timing. There are two very special proteins that he discovered, but actually we don't yet know how they work in a precise way. So a lot of what I'm doing now is, with other colleagues at Yale, is trying to understand almost at an atomic level, and we will succeed over the next 5 or 10 years, to understand exactly how the precise timing in the brain is achieved. Now, my belief is when we understand that, we'll also have the means to understand another level of sophistication, ultimately, of the cognitive process. I can't prove that today, but I believe that that's true. So I do think there are probably important elements. I'm also starting a laboratory at University College London. We have a program at Yale called the Yale-UCL Collaborative.
Okay.
At UCL, they call it, by the way, the UCL-Yale Collaborative. They're probably both correct. And this enables Yale professors to spend some significant time in London at University College London to take advantage of what they have to complement what we have, and also vice versa. And so I'll be working in the Institute of Neurology at UCL, which is really the world leader in the genetics of human neurological diseases. And my clinical colleagues there are very much of the persuasion that there are many common and rare neurological syndromes that are so-called, they call them synaptic disorders, in which the very proteins that we study are likely to be altered in the basis of these diseases. And we're just really at the tip of an iceberg in terms of understanding how, what the disease impact is in psychiatry and in neurology of the discoveries that my colleagues and I have made. are. And so that's another ambition I have, is to try to see that through, and that is something I'll be doing in London primarily.
And so obviously you'll be spending quite a bit more time there.
Yeah, I will be.
Excellent.
Yeah.
Jim Rothman, this has been a real pleasure. Thank you so much for spending some time with us and sharing your thoughts.
Thank
and
you,
we
Eric.
look forward to the results of the work to come.
Thank you, Eric.
Thanks very much to all of you for watching at Yale Live. Please join us again next time.